Sleep. Some of us get way too much, others a different story. But as university students, we probably tend towards the latter. We all need it though. This includes humans, fish, dogs, birds, and even horses who stand up sleeping.
Sleep deprivation can be understood as simply not getting enough sleep. An immediate effect of not getting enough sleep is the loss of focus, impaired memory, and the inability to respond to normal stimuli. Sleep deprivation does not only affect individual, but it also affects the community safety. The most important component of sleep is of course our brain, and one mechanism that ensures the protection for any harmful substance is the brain-blood barrier. The blood-brain barrier is a layer of tightly packed endothelial cells that surround the capillaries of the brain. It is responsible for allowing some molecules into the brain while preventing others, potential harmful factors from entering. Hence, it is critical for protecting the brain. The blood-brain barrier is highly selective semi-permeable border that allows the passive diffusion of water, gases, lipid-soluble molecules, and selective transport of other essential molecules such as amino acid and glucose. This also prevents the entry of harmful factors such as pathogens, toxins, and drugs. It has been shown in a study done in 2004 that a direct correlation between chronic sleep deprivation and the function of the brain-blood barrier. The study found that chronic sleep deprivation decreased the transportation function of the brain-blood barrier, affecting the function of the glucose transporters and reducing vascular sensitivity in the brain.
Our brain is the command center of our central nervous system. It is comprised of thousands and thousands of neurons, dendrites, axons, and neuroglia. Neuroglia are also called glial cells. They are non-neuronal cells in the central nervous system comprised of the brain and spinal cord, and the peripheral nervous system. We might also know them from our BAPS 1201 lectures. Their role is to provide protection, structure, and nutrients to the neuron, as well as form the myelin. There are 4 different types of neuroglia: astrocytes, microglia, oligodendrocytes, and Schwann cells. In order to understand the relationship between sleep and our brains, we will focus mainly on astrocytes and microglia. Astrocytes are the most abundant type of glial cells in the central nervous system. They also serve as a functional and structural link between neurons, non-nervous tissue, and extracellular fluid in the maintenance of the blood-brain barrier. They are also tasked with the role of axon guidance, synaptic support, provision of nutrients to the nervous tissue, maintenance of extracellular ion balance, and a role in the repair of the brain and spinal cord following traumatic injuries. The other type of neuroglia, microglia, serves to mediate immune responses in the central nervous system by acting as macrophages, clearing cellular debris and dead neurons from nervous tissue through the process of phagocytosis, also known as cellular eating. Microglia also take part in a pruning process during synaptic development. They tag unwanted synapses through to receptors in the white blood cells. Moreover, sleep deprivation causes an increase in immune response to microglia. When the human body is chronically sleep deprived, there is an accelerated activity of phagocytosis. This causes the brain to be in an inflammatory state, making it more susceptible to damage. The astrocytes begin to be more active between the synapses of neurons, as they need to clear areas of glutamate and potassium ions. This was all demonstrated by a study by Belzi et al. As they conducted an experiment on mice and found that astrocytic activity becomes unregulated and essentially goes into overdrive. This study observed the changes in neuroglia in the brain of 4 groups of mice that were kept awake for different periods of time. The mice which were sleep deprived showed that the astrocytes began to engulf organelles of axons, in other words, eating them. This was all done through an electron microscope which observed the cortex of mice, and their behavior was also observed through an installed camera. Unfortunately, after the experiment, the mice were killed. The study also found that sleep loss may trigger a phagocytic response in sustained neuronal activity. The scientists also observed that cerebral spinal fluid inflammation was present due to microglial activity in a severely sleep-deprived brain. Therefore, chronic sleep deprivation can cause an upregulation of neuroglia, especially microglial cells, which can lead to neurological damage, or in simple terms, eating of the brain. So what's the take-home message? It's simply to sleep more. Sleep is not for the weak. Sleep will make you weak.